today in this video we are going to discuss about meiosis. In last video we have completed about mitosis, now we are going to see about meiosis. So in case of mitosis there is only one division, but meiosis there are two divisions, meiosis 1 and meiosis 2. So in case of meiosis 1, the same all the phases will be repeated, G1, S, G2, karyokinesis. In karyokinesis we have prophase 1, metaphase 1, anaphase 1, telophase 1, and once again the prophase 1 is divided into, and only prophase 1, not prophase 2. Okay, so only prophase 1 you have to study, so you have to thank meiosis. You have to thank meiosis or not? Yes, why? Because these many stages are occurring only once. So, either way, that prophase 2 we have to study a lot. Okay, so it made our process very simple. So, first we will thank for that. Only in prophase 1 we have subdivisions. What are the subdivisions? Leptoten, zygoten, packeten, diploten, diakinesis. This is only seen in prophase. In the same phases when the prophase 2 la irukka. The prophase 2 is very simple. Okay, and metaphase 2, anaphase 1, telophase 1. After this, after karyokinesis, we have cytokinesis. We have what? Cytokinesis. So here is the simplified structure. So meiosis 1, meiosis 2. Meiosis is divided into meiosis 1 and meiosis 2, but mitosis is not like that. Mitosis is only single short division. Okay. So here in meiosis 1, karyokinesis 1, what we can write under that? Yes, P mat. We can write P mat. In that prophase, how many subphases? 5. So what is the key to remember? Lazy pack. Lazy pack. Why? Because however you study, evlo time ninga padichalu, you will feel like you confused. Okay. So how to remember that? Leptoten, lazy, La for leptoten, Zy for zygoten, P for packeten, pack for packeten. Then we have diploten, PP, diploten and diakinesis. Okay. After this, the cell will divide into 2. At the end of meiosis 1, at the end of meiosis 1. Okay, before karyokinesis, is there any process? G2, yes, G1, yes, G2. After the interphase only we have karyokinesis. Okay, so after this we have karyokinesis 1. Karyokinesis 1 is divided into 4 subphases. Adila prophase is divided into 5 subphases. Okay, now after cytokinesis we get 2. Yes, 2 beautiful. Okay, 2 beautiful daughters and now. Daughters. If it is if it occurs in female body, then daughters. If it occurs in male body, sons. Okay, but anyhow father can contribute to daughter also. If it is X sperm, then it will be daughter. If it is Y, it is son. Okay, so in case of meiosis 1, at the end you have 2 products, 2 cells. Now these 2 cells will undergo once again G1, G2, not S phase. Not S phase, and this phase is like very short, very short. That's why we are calling this as not interphase 2. We are giving a new term for that, that is called interkinesis. This is very, very important. Usually G1, S, G2 in the 3 phase you நம்ம என்ன சொல்லுவோம் interphase அப்படினு சொல்லுவோம். இங்க meiosis 1 ல இருந்து meiosis 2 க்கு நடக்கிற transitionல நடுவுல இருக்கிற G1, G2 நம்ம என்னன்னு சொல்றோம்? Interkinesis. After G1 and G2 we have karyokinesis 2. Karyokinesis 2 la once again prophase 2, metaphase 2, anaphase 2 and telophase 2. Is that clear? After this, how many cells are undergoing the process? 2 cells, because the product of meiosis 1 is 2 cells. So obviously inga rendu cell lume indha ella process me nadakudhu. Interkinesis rendu cell la nadakudhu, prophase 2, metaphase 2 ellame rendu cell la nadakudhu. So at the end you will get 4 cells. At end you will get 4 cells. Is that clear? So meiosis, yes, what doubt you have? Okay, at the end of meiosis 2 we will have how many cells? 4 cells. At the end of meiosis 1 we will have 2 cells. Okay, we will see that whether it is haploid or diploid, everything we will read. Okay, so now first slide. Okay, In first slide, prophase 1, metaphase 1, the number is new. Okay, this number you didn't study in mitosis. In the number meiosis la padikringa. Mitosis la just P mat appdin padichom. Okay, next. What is interkinesis? Transition between first division to second division without S phase. Okay. Okay, now most of your seniors will make mistake in this. மியோசைட் அப்படின்றத பார்த்துட்டு ஓகே அது மியோசிஸ் தானே கண்டிப்பாக அந்த செல் வந்து ஹாப்ளாய்டாக தான் இருக்கும் அப்படின்னு சொல்லிட்டு அவங்களே ஒரு கெஸ் பண்ணி ஆன்சர் எழுது. பட் தட் வில் பி ராங். மியோசைட் இஸ் த செல் விச் அண்டர்கோஸ் மியோசிஸ். வாட் இஸ் மியோசைட்? Maybe I would have told faster. Site means cell. Site means what? Cell. Myo means meiosis. Myo means meiosis. So the cell which undergoes meiosis is called myocyte. Okay, oocyte அப்படினா the cell which undergoes oogenesis is called oocyte. Okay, cyte means cell, myocyte means cell which undergoes meiosis, and it is diploid. It is diploid. In case of human beings it's 2n. And what is the meaning of 2n? What is the meaning of 2n? It contains 46 chromosomes. How many chromosomes? 46 chromosomes. Okay. 46 chromosomes. If suppose the cell completed S phase, now we are taking the myocyte which completed S phase. இதுதான் மோஸ்ட் ஆஃப் த ஸ்டூடெண்ட்ஸ்க்கு வந்து நீங்கள் கன்ஃபியூஸ் பண்ணுறது இந்த குரோமாட்டிடோட கவுண்ட் தான். ஏன் குரோமாட்டிடை வந்து 46ன்னு எழுதாமல் we will write 92 chromatids. எவ்வளோ chromatids எழுதுவோம்? 92. ஏன் 92 chromatids எழுதுறோம்? 46 குரோமோசோம்ஸ்க்கு இப்போ இது குரோமோசோம் நம்பர் 1 அப்படின்னா இதுக்கு ஒரு குரோமாட்டிட் இருந்தால் தான் அது ஒரு குரோமோசோம். பட் வை வி ஆர் கவுண்டிங் குரோமாட்டிட் as 2? Because it completed S phase. It completed S phase. So after S phase it develop another one copy of chromatid because new copy of DNA is present there. புரிஞ்சுதா? So that's why for 46 chromosomes we are counting it as 92 chromatids. இது புரிஞ்சுதா? Why 92 chromatids? Because the cell completed S phase. ஒருவேளை அந்த செல் S phase G1 phase அப்படின்னு question கேட்குறாங்க. G1 இருந்ததுன்னா 46 chromosome, 46 chromatid. And one more doubt you all have, whether you have to count the chromatid like this 1 2 3 4 இப்படி count பண்ணுமா? No, that is wrong. You should not count like that. You should count like this is one chromatid and this is another chromatid. So only 2 chromatids. Okay. Okay, now at the end of meiosis 1, this cell will have 23 chromosome. How many chromosome? 23 chromosome, and here also 23 chromosome. So chromosome is split into 2. So 46 chromosome half ஆதா வருது 23. அதனால நம்ம 2n நம்மளும் என்னவா ரெடியூஸ் பண்ணிட்டோம்? n. This is called reductional division. This is called reductional division because the initial parent cell had 46 chromosomes, but while giving it to daughter or son it showed a partiality. What it did? 46 chromosomes அப்படியே கொடுக்காம 23 ரெண்டு பேத்துக்கும் half தான் கொடுத்துருக்காங்க. Like your parents are buying one new eraser and they'll cut into 2 half and they'll give இது உனக்கு. When you ask question what they will say? எப்படி கொடுத்தாலும் தொலைக்க தான் போகிறேன், அதனால என்ன பண்ணேன், ஃபஸ்ட் இது தொலைச்சிட்டு அதுக்கப்புறம் ஆஃப்டர் தட் ஐ வில் கிவ் யூ another half eraser. Same way here meiosis also what it is going to do, 46 chromosomes are 23 chromosomes and 23 chromosomes. But what will happen to the number of chromatid? That is very important. What will happen to the number of chromatid? Chromatid will be 46. Chromatid will be 46. So 46 chromatid and 46 chromatid. Okay. Why 46 chromatid? Because each chromosome looks like this. Okay, so 23 chromosomes means how many chromatid will be there? Into 2, 46. Idu purinjudha? Why 46? Did you understand why 46? Any doubt? No. Okay, now what will happen in meiosis 2? Here I have written meiosis 2. What will happen? Now this will split. Where you can split? 23 chromosomes. So adile innu decrease panna mudiyadhu. 23 chromosomes and 23 chromatid. So this 46 can be divided by 2 in the next division. Yes or no? So upcoming cells ku 23 chromosome and 23 chromatid. You have any doubt? Yes, here, see, yes, very good. Here only splitting of centromere will take place. Here only splitting of centromere. Before that, there is no splitting. There is no splitting. For example, if this is one chromosome, in the mari 46 chromosomes irukku. In the 46 chromosomes, in the pen na cut panave illa, 46 pens irukku. So 23 pens or boys ku 23 pen girls ku na kudukren. Next, what I am going to do? I am going to cut the pen into another half. So once again I will get 23 piece and 23 piece from each pen. So now I will give to another half people. Okay. So that is the reason why we are getting 4 cells. Okay. Because each will get 23 chromosome and 23 chromatid. How many of you didn't understand this? You didn't understand. Anyone else? Anyone? Okay. See here, this is chromosome 1, chromosome 1. Like this, we have 46 chromosomes. We have how many chromosomes? 46 chromosomes. The first division is like this. In the first division, I am going to divide 23 chromosome and another 23 chromosome. First division இப்படி இருக்கு. புரிஞ்சுதா? 46 pens இருக்கு. 46 pensல 23 for boys, 23 for girls. Now, in the next division, what I am going to do? Suppose I write the same chromosome here. Like this, I have 23. Now, only I have 23. So, in the next division, I will cut the centromere. I will split the pen. I will break the pen into 2 pieces, and once again I will give 23 half pen and 23 half pen for another group. புரிஞ்சுதா? தனுஜா? Okay. Now, that's the reason we are getting 4 cells. So here you have to write 23 chromosome. 23 chromosome and how many chromatid? 23. Hemavati, how many chromatid? 23. How 23? Whether parent is affordable, parent nala 23 kudukka mudiyuma? Yes, because parent is having how many chromatid? 46 chromatid. So adanal avangita 46 irukku. Adanal avanga each daughter ku avanga evlo kudukalam? 23. In the parent kit 46 chromatids irukku. Adanal for each 23. Already chromosome 23 da irukku. Adanal split panni kudukranga. புரிஞ்சுதா? chromosome just 23. அதனால அதை split பண்ணாம கொடுக்க முடியாது. So they are going to split the chromosome into 2 equal halves and they are going to give for their daughter cells. Is that clear? Clear? That's why yes, haploid. And this to this, the division is called equational division. Listen. This to this, the division is called equation. Why? Because n, from n we are getting once again n. So the division is called equational division. Okay. Next we are going to see the actual subphases. Leptotin. The first phase is leptotin. So before that, some short question answer session. What is interkinesis? Okay, Monica. Yes, can you come and explain what is the difference between interphase and interkinesis? You have to write here the difference between interphase and interkinesis. Right. Okay. Okay, clap everyone. This is correct, right? Yes. So this is the difference. This is the difference between interkinesis and interphase. Because in interkinesis there is no S phase, but in interphase there is a S phase. Okay. Next. Here we will see about leptotin. So listen carefully. Next I will ask question from this leptotin. Okay. In case of leptotin, this is prophase. Usually நம்ம படிச்ச prophase-ல நம்ம என்ன படிச்சிருப்போம்? நியூக்ளியர் envelope வந்து disappear ஆயிடும், centriole வந்து poles-க்கு வரும், அதுக்கப்புறம் செல் ஆர்கனல்ஸ் எல்லாம் டிஸ்அப்பியர் ஆகிடும். ஏன்னா செல் டிவைட் ஆகும்போது இதெல்லாம் டிஸ்டர்பன்ஸாக இருக்குது அப்படின்னு சொல்லிட்டு அதுவே எல்லாத்தையும் டெர்மினேட் பண்ணிடுது. அப்படி தான் நம்ம ப்ரொஃபேஸ் படிச்சிருக்கோம். பட் இந்த ப்ரொஃபேஸ் அந்த மாதிரி கிடையாது. இந்த ப்ரொஃபேஸ் கொஞ்சம் வேற மாதிரி. ஓகே, எப்படி வேற மாதிரி அப்படின்னா ஆல் த ஆக்சுவல் ப்ரொஃபேஸ் ஸ்டேஜஸ் எல்லாமே லாஸ்ட்ல தான் நடக்கும். லாஸ்ட் அப்படின்னா என்ன ஸ்டேஜ்? டயாகைனசிஸ். லெப்டோடென், ஜைகோடென், பாக்கெடென், டிப்ளோடென், டயாகைனசிஸ். ஸோ ஒரு ப்ரொஃபேஸ்க்கு இருக்கிற எல்லா கேரக்டருமே எங்கே எக்ஸ்பிரஸ் ஆகும் அப்படின்னா டயாகைனசிஸில் தான் எக்ஸ்பிரஸ் ஆகும். புரிஞ்சுதா? ஸோ ஃபஸ்ட் இருக்கிற ப்ரொஃபேஸ் வந்து அதனுடைய ஃபீச்சர்ஸை மட்டும் தான் நம்ம படிக்க போகிறோம். ஸோ அது ஆஸ் யூஷுவலாக ஒரு செல் எப்படி இருக்குமோ அப்படி தான் இருக்குது. யூ கேன் see here nuclear membrane is there and chromatin also like same like chromatin. Just what happens? Compaction happens. What happens? Compaction, so that it will be easy for pairing. Okay, so here all the maternal and paternal chromosomes are going to talk with each other. Okay, sometimes maybe in our home father and mother because of fight they may not talk to each other, but in our sex organs, be it ovary or testis, always this maternal and paternal chromosomes will gossip about each other. Hey, what you had for lunch? Hey, what, how are you Manny, how are you D, like that. This both will, okay, this maternal and paternal chromosomes, both the chromosomes are going to talk with each other. Okay, so for that they need some kind of preparation. So we are going to see that. First is leptoten. First in leptoten it's going to do some kind of makeup. Listen here. In leptotens these chromosomes are going to do some makeup because they want to arrange themselves, because they are going to meet their pair. They are going to meet their pair. Where they are going to meet their pair? In the next phase. In the next phase. Which is next phase? Yes, in the zygote end they are going to meet their pair. The maternal chromosome will meet the paternal pair and the paternal pair will meet the maternal pair. Okay, so here what happens? The nuclear membrane is intact, so no one will disturb the nuclear membrane. Okay, because inside chromosomes are doing makeup. Okay, inside chromosomes are busy, they are dressing themselves very nicely. Because why? Because they are going to meet their pair. Okay, so they are going to dress them nicely, and so that process is happening inside. So there is no disturbance for nuclear membrane or cell organelles or centrioles. There is no aster ray formation because they don't know that someone is going to separate they both. Okay, so now they are happy. Okay, next chromatin attaches to nuclear membrane. So they are getting attached to nuclear membrane like a bouquet. So this is a keyword. You can remember the leptoten is equal to bouquet stage. Okay, it is similar to bouquet stage and mostly attached to one end, because all the chromatin fibers will be attached to one end. Which end? That is the nuclear membrane. They are attached to nuclear membrane. And why I made here yellow dots? Yellow dots? Yellow dots? Nucleosome. Yellow dots are nucleosome. Okay, next chromatin appears as beaded. Yes, because chromatin will look like bead structure. Okay, that beads are called, Nithya, listen, beads are called nucleosomes. You write later, I will give you time to write, don't write now. Okay, okay, maybe you know who is homologous pair, but many of you, maybe in the class itself, you don't know what is homologous chromosome. How many of you don't know what is homologous chromosome? How many of you don't know what is homologous chromosome? Okay, you don't know. Okay, listen. So in each and every cell we have 46 chromosomes. How many chromosomes we have? 46 chromosomes. And we came from single cell. You agree? We came from single cell. What is the name of the single cell? Zygote. How the zygote formed? Okay, fusion of gametes. So the egg cell contains 23 chromosomes met with the sperm which contains 23 chromosomes. So ஒரு egg 23 chromosome இருக்கிற egg because எப்பவுமே half வந்து half ஆவே இருக்காது. நேச்சரே எந்த ப்ராசஸ் நடக்க வைக்க ட்ரை பண்ணுனா நேச்சர் வந்து always will try to make haploid as a diploid. Haploid as a diploid. Haploidனா என்ன, diploidனா என்ன? ஹாப்ளாய்ட்னா ஹாஃப் செட் ஆஃப் குரோமோசோம். இப்போ அது வந்து 23 தான் இருக்கணுமா அப்படின்னா நோ. இப்போ ஃபார் எக்ஸாம்பிள் சில ஆர்கானிசத்துக்கு மொத்தமாகவே 8 குரோமோசோம் தான் இருக்குது அப்படின்னா அதில் 8 வந்து டிப்ளாய்ட், 4 வந்து haploid. So irrespective of the chromosome number, half set வந்து நம்ம என்னன்னு சொல்றோம் haploid, full set வந்து diploid. Okay, so in case of this sex cells, yesterday only I taught you which is sex cell. I asked which is sex cell. Sex cell. Yes, egg and sperm. Egg cell and sperm cell. Which is sex cell? Egg cell and sperm cell. Okay, this egg is a egg, sex cell, and it is sperm. Now, if you have doubt, see, egg வந்து ஒரு 23 chromosomes எடுத்துட்டு வருது. நானும் வச்சிருக்கேன் அப்படி சொல்லிட்டு பேக்கில் 23 chromosomes வச்சிருக்கேன். நான் சும்மா வரல, நான் எவ்வளோ chromosomeஸோடு வரேன்? என்னோட பேபிக்கு நான் எவ்வளோ கொடுக்க போகிறேன்? 23 chromosomes. அதில் 22 chromosomes வந்து 22 plus 1 நம்ம டிவைட் பண்ணுறோம். எப்படி டிவைட் பண்ணுறோம்? 22 plus 1. Why we are dividing 22 plus 1? Because this 22 is called autosomes. This 22 is called autosomes. அது என்னது ஆட்டோ காரு? ஆட்டோனா என்ன? Autosomes are the chromosomes which control your body characters. Which control your body characters. Okay, many times we look at mirror and we will think, எனக்கு இன்னும் கொஞ்சம் hair கொஞ்சம் நல்லா இருந்திருக்கலாமே, எனக்கு இன்னும் skin tone இன்னும் கொஞ்சம் white-ஆ வந்திருக்கலாமே. Yes or no? இந்த மூக்கு கொஞ்சம் நல்லா இருந்திருக்கலாமே. Yes or no? We will, no, I used to see and I used to think always, இன்னும் கொஞ்சம் அழகா இருந்திருந்தாதான் என்ன, like that, and why I became like this. So we will never appreciate that, oh my god, I have 2 eyes, oh my god, I got a very good nose and I got a teeth and I am healthy. No, we will never appreciate. We always see the flaws in us. The reason for our body is because the 22 chromosomes are designing your body. Okay, your body design is made by 22 chromosomes. So only 22 chromosomes? No, in the 22 ku or role irukku. Then another 22 is carried by the sperm. Now sperm ஒரு பேக் எடுத்துட்டு வருது. அதில் அது என்ன பண்ணுது? அது ஒரு 22 chromosome plus 1 chromosome. ஸோ அதுவும் என்ன சொல்லுது? நீ மட்டும் எப்படி பேபிக்கு கேரக்டர்ஸ்லாம் கொடுக்கலாம், நானும் கொடுக்குறேன், அது நம்மளோட பேபி இல்லையா? ஸோ அப்படி சொல்லிட்டு paternal chromosomes. These 22 chromosomes are called paternal chromosomes because they are coming from father. Okay, another parent. Okay, that's why it is called paternal chromosome. Now this 22 also called autosomes. ஏன் autosomes? Because இதுவும் அந்த பேபியோடைய, இந்த சிஸ்டர் குரோமாட்டிட் கிடையாது, அந்த பேபியோடைய ஸ்கின், ஹேர், இது எல்லாமே கண்ட்ரோல் பண்ணக்கூடியது இந்த ரெண்டு 22 plus 22 how many chromosomes? 44 chromosomes சேர்ந்து டிசைன் பண்ண ஒரு டிசைன் தான் நீங்கள் எல்லாருமே. ஓகே, 44 chromosomes-உம் சொல்லுது இப்படி தான் இருக்கணும், இந்த இடத்துல தான் இருக்கணும் அப்படி சொல்லிட்டு, இவ்வளோ தான் ஹேர் லென்த் இருக்கணும், இவ்வளோ தான் வந்து பாடி ஷேப் இருக்கணும், இவ்வளோ தான் ஒபிசிட்டி இருக்கணும். ஸோ எவ்ரி திங் இஸ் கண்ட்ரோல் பை யோர் chromosomes. நெக்ஸ்ட் இந்த 1 chromosome, we should not take this lightly. Yeah, this only brings a smile in the parents' face. Yes or no? Some parents wish I want girl child. Some parents wish I want girl child. Many parents wish I want boy child. That girl or boy, that sex of the child is decided by this 23rd chromosome. This is called 23rd chromosome. Now this chromosome is called sex chromosome. This is called not sex cell. This is called sex chromosome. In 23 chromosomes we have flavors. 22 controls your body character. The last one controls the sex of the baby. Okay, so sex of the baby is controlled by the first chromosome, and 23rd chromosome and 23rd chromosome. Okay, so now this 23 is present only in sex cell. But what about our normal skin cell? In normal skin cell we have 46 chromosomes. We have 46 chromosomes. So the meiocyte also. Now we are talking about meiocyte. Meiocyte also have 46 chromosomes. This 46 will now pair as 23 pairs. 46 will become how many pairs? 23 pairs. 23 pairs. Okay, so 23 pairs means chromosome number 1 of mother will meet chromosome number 1 of father. Okay, so now these both Now my skin cell, you are just studying, I gave each and everyone my skin cell. எல்லாருக்கும் என்னோட skin cell ஒன்னு கொடுத்துட்டேன், மைக்ரோஸ்கோப் கொடுத்துட்டேன். நீங்க மைக்ரோஸ்கோப் கீழ என்னோட skin cell பார்க்கறீங்க. 23 குரோமோசோம்ஸ் வந்து நல்ல ஹேர்லாம் வச்சு மல்லிப்பூலாம் வச்சு சாரிலாம் கட்டி நல்லா டிசைன் பண்ணி நல்லா இருக்கு. அந்த 23மே என்னோட மதர் கொடுத்த மெட்டர்னல் குரோமோசோம்ஸ். இன்னொரு 23 குரோமோசோம்ஸ் நல்லா ஒரு சொட்டையோ ஸ்பெக்ஸ்லாம் போட்டுட்டு பேண்ட் ஷர்ட்லாம் போட்டுட்டு ரொம்ப பார்க்கறதுக்கு ஃபுல்லாக மேலாக இருக்குது. அது வந்து பெட்டர்னல் குரோமோசோம். அது வந்து என்னது? பெட்டர்னல் குரோமோசோம். இப்போ நீங்கள் என்ன பண்ணுறீங்க, இது ரெண்டுத்தையும் பிடிச்சி மேட்ச் பண்ணுறீங்க. ஓகே, ஒரு ஃபீமேல் ஒரு மேல், ஒரு ஃபீமேல் ஒரு மேல். இதுதான் ஹோமோலோகஸ் குரோமோசோம். திஸ் இஸ் கால்ட் homologous chromosome. Now the chromosome 1 of mother is matched with chromosome 1 of father. Now these both one are called homologous chromosomes. These both are called homologous chromosomes. Okay, now இந்த chromosomes இருக்கு இல்லையா, இந்த chromosomes-க்கு ரெண்டு chromatid இருக்கு. எத்தனை chromatid இருக்கு? ரெண்டு chromatid. இந்த ரெண்டு chromatid-உமே sister chromatid. அதாவது அம்மா குரோமோசோம்ல ரெண்டு கை இருக்கு, அது ரெண்டும் அந்த அம்மாவுடைய தான். ஸோ அதனால அது ரெண்டும் சிஸ்டர். ஃபாதர் குரோமோசோம்ல இருக்கிற ரெண்டு ஆர்ம்ஸ் வந்து இட் பிலாங்ஸ் டு பெட்டர்னல் குரோமோசோம், அதனால அது ரெண்டும் சிஸ்டர். பட் நவ் வாட் இஸ் கோயிங் டு ஹேப்பன், திஸ் ஆர்ம் இஸ் கோயிங் டு எக்ஸ்சேஞ்ச் த இன்ஃபர்மேஷன் வித் திஸ் ஆர்ம். நவ் தீஸ் போத் ஆர் நான் சிஸ்டர் குரோமாட்டிட்ஸ். தீஸ் போத் ஆர் நான் சிஸ்டர் குரோமாட்டிட்ஸ். இப்போ மெட்டர்னல் குரோமோசோம்ஸ் எல்லாம் மெட்டர்னல் குரோமோசோம்ஸ்டே பேசிக்கிறதுனால எந்த யூஸும் இருக்க போகிறது இல்லை. ஸோ மெட்டர்னல் இஸ் கோயிங் டு காசிப் வித் paternal chromosome. Now this both are called non-sister chromatids. So in exam you have to check clearly whether the crossing over occurs between sister chromatids or non-sister chromatids. You have to check two words. One is homologous chromosome. Yes or no? Crossing over will occur between 100% sure it will occur between homologous chromosome. What is meaning of homologous chromosome? Pair. Okay, and between the homologous chromosome, between non-sister chromatids, between the non-sister chromatids. How many of you didn't understand what is homologous chromosome? Everyone understood what is homologous. Okay, next is zygoten. Next is zygoten. In case of zygoten, you arranged. Okay, now I gave you what cell? My skin cell. I gave you my skin cell. So என்னோட skin cellல இருந்து நீங்க வந்து 46 chromosomes 23 pairs ஆ மாத்திட்டீங்க. ஜோடி நம்பர் 1 any season like a pair. Okay, now 23 chromosomes are standing in a pair. Okay, 23 pairs இருக்கு. Next அவங்க என்ன பண்ண போறாங்க அந்த pair அப்படினா they are going to exchange the information. They are going to exchange the information. For that first they are coming closer. First they are coming closer, because you know always like our parents, they are not close. They are not close, always fighting, always fighting. So first in the zygoten phase, they won't come themselves closer. So you are making them come closer. Okay, so the maternal chromosome 1 will come closer to paternal chromosome 1. Okay, that's what I have written. Homologous chromosomes start pairing. They will start pairing. This process is called synapsis. This process is called synapsis. Now you understood what is synapsis? Yes, what is synapsis? Pairing of not non-homologous chromosomes. Okay, homologous chromosomes. Okay. Next, the synaptonemal complex is formed. What is that synaptonemal complex? Synaptonemal complex means they both are saying, suppose Preeta Sri is taking chromosome 1, okay, of mother and father. She catched my maternal and paternal. Now these both are saying that ஐயோ எனக்குலாம் கூட இருக்கிறதுக்குலாம் பிடிக்கல, நாளா பேச மாட்டேன், ஐயோ the character is like that. They both are fighting always. Okay, now Preeta Sri has to apply some gel. Apply some gel. See whatever she talks also, she likes you so much. See whatever he does with you also, ultimately at the end of the day he will support you. Like that she has to apply some gel between both the chromosomes. That is called synaptonemal complex. That is called synaptonemal complex, which is going to attach the non-sister chromatids, which is going to build the bridge between non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. So what is that exactly? A protein. That is a protein. A protein lattice, lattice means the arrangement, okay. A protein lattice that keeps homologous chromosomes. Maybe Preetha Shri வந்து ஒரு பொய் சொல்லியிருக்கலாம், அவங்களுக்கு உங்களை ரொம்ப பிடிக்கும், உங்களுக்கும் அவங்கள ரொம்ப பிடிக்கும் அப்படின்னு சொல்லி. பட் அந்த பொய் தான் அவங்க ரெண்டு பேர்த்தையும் it's going to keep them together, okay. That is called synaptonemal complex. Understood? What is synaptonemal complex? It's a kind of protein which is going to gel both the chromosomes together, but still they both didn't start to exchange the information, okay. They need some time, okay. After some time father has to give the proper explanation, then only the mother will start yes, exchanging the information, okay. Now you have arranged, right, all the jodis you have arranged 1 la irundhu 23 அழகா pair நீங்க arrange பண்ணிருக்கீங்களா. Now this each and every pair is called dyad or bivalent or homologous pair. These three are the different words given to the same structure, same pair. ஒரு pairக்கு நம்ம கொடுக்கிற different names, dyad, bivalent, even another one name also you can add, it is also called tetrad. It is called tetrad, okay. But what is the difference between bivalent and tetrad? Bivalent means the structure will be like this. Suppose if yes, this is 2 chromatids of 1 chromosome and this is 2 chromatids of 1 chromosome. This is chromosome 1, chromosome 1, chromosome 1, chromosome 1. 1 1 oda da pair agum, 2 2 oda pair agum, 3 3 oda pair agum, like that 23 will pair. Okay, now இந்த மாதிரி ரெண்டு chromosomes பக்கத்து பக்கத்துல நிக்கும்போது we are getting a structure like this. This is called bivalent. Oh, after this no, okay. A tetrad அப்படினா then see first they both are like very rigid, okay. They are making some scene. I will not talk. After some time no one can stop them, okay, like that. So this structure, when they started to talk with each other, this structure is called tetrad. This is called tetrad. Actually they started to exchange the material, but in case of zygote it will look like this, but if you see from the down, actually there is no crossing over, only synaptonemal complex is there, only the whatever the lie Preetha Shri told, no, that lie is running between both the chromosomes, but actually these chromosomes are not paired, okay. They didn't exchange any kind of information, there is no shuffling, okay. So far clear. So you can see here nuclear membrane is intact. We are in which phase, Nityashri? Zygote comes under which phase? Prophase 1. Prophase 1 comes under which phase? Inside meiosis 1? Inside meiosis 1? Karyokinesis. Karyokinesis number 1, okay. Why you are so dull? Okay, next I will take your name as example, okay. Packetin, okay. Next is packetin, okay. So in packetin you imagine totally one round tables are there, okay. 23 tables irukku, round tables. Hey, listen, 23 tables irukku, 2 chairs, this side maternal chromosome, that side paternal chromosome, talking, they are started to exchange their information, okay. That process, the exact crossing over takes place at which stage? Packetin stage, okay. In packetin stage both are packed, okay. So tetrad clearly visible, meaning that you can see that they were laughing ha 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 and they were talking, okay. That tetrad is clearly visible. And then recombination nodules appear. Recombination nodules means you can see the places where the exchange actually happens. Okay, here doesn't need, right? Okay, suppose if this is the chromosome 1 and this is 1 arm, and now this is 1 chromatid, this is the chromosome 1 from okay. So like this, now these both, no, they started to exchange the information, and this is called site of crossing over. This is called site of crossing over. It is also called as chiasmata. This is called what? Chiasmata, okay. And recombinase is an enzyme that is going to mediate the crossing over, okay. So crossing over mediate பண்ணக்கூடிய enzyme பேர் என்ன? Recombinase. So recombinase ஓட work என்ன அப்படினா, இப்போ வந்து before they talk to each other they are pure, okay. if you want, you would have biased them. I want to go for a tour in my school, so you give me 5,000 rupees. நீ என்ன சொன்னாலும் அவங்க ஒருவேளை கேட்டிருந்திருப்பாங்க because they were pure. But after crossing over, some of your mother character entered to your father brain and your father character entered to mother brain. Now they will behave like onion. Okay, if you talk means first they will say okay, then they will say I will ask your mom, then they will say not okay. Okay, because both exchanged the information and now both got shuffled. So now you are getting confused, who is my mother, who is my father. Okay, because they both are shuffled. Idu mattum da puriyudhu, concept puriyala. Okay, they both are what? Shuffled. Okay, so that is next. So, okay, they were talking each other and they have exchanged the information, and after exchanging, no, if you want to see the difference, so I can show some yellow character came to this chromosome and some white came to this yellow. So, now little character they have exchanged. Okay, they have exchanged information and their brain got polluted. Understood? Okay. After this what will happen, then once again you are saying that okay பேசுறதெல்லாம் போதும் ரெண்டு பேரும் வாங்க அப்படி சொல்லிட்டு you are going to separate the pair, homologous pair. அப்போ கூட they are not coming away from each other, they are joined at one place. That place is called chiasmata. So they literally they were like this. Okay, like this. Okay, they were like this. illa na varamaten vitru solranga, but you are pulling them away from each other. This is called diplotin stage. Here terminalization starts. அவங்க வந்து ரெண்டு பேரும் செப்பரேட் பண்ண ஆரம்பிக்கிறாங்க, but still they are not completely separated from each other. Just the process begins. Okay, and dissolution of synaptonemal complex. Preetha Shri சொன்ன போயெல்லாம் இங்க என்ன ஆயிடுச்சு? Dissolution means it's going to dissolve. So, and both the pair are going to separate from each other except at the sites of chiasmata, meaning except this site. அவங்க எங்க exchange பண்ணிருக்காங்களோ, suppose like this, if it is like this, suppose previously they are like this, now they are like this. Okay, except the site, other places have dissoluted. Okay, now next what will happen? Next the process is going to end. So terminalization of chiasmata. So chiasmata also completely dissoluted. Terminalized means end, and chromosomes are fully condensed, meaning now you got the perfect chromosome structure. Previously I have drawn chromosome structure, but it is not like that. Now only full condensed chromosome you are getting, meaning from the chromatin, this line meaning is from the chromatin you are getting full condensed chromosomes. Okay, next is spindle assembly. Now only actual prophase you can see. Nucleolus disappear, nuclear envelope breaks down, and centriole move towards the poles. So you can see the late prophase figure here. Okay, so the spindle fibers formed, and the spindle fibers are going to attach with chromosomes, attach with kinetochore of the chromosomes. Okay, so till this we'll stop here, and tomorrow in the next class we'll continue the next part. Thank you.